Okay, hi, thank you for coming. My name is Igor Bogićević and I'm CTO and one of the co-founders of Genomics. So, just to tell a little bit about us. So we were founded in 2009, 2010, and we're actually based in Cambridge, Massachusetts, London, and Belgrade. So, just to give you a brief history of the company, so the other co-founder, Dennis Kural, who is the CEO of the company, is actually one of the original members of the 1,000 Genomes project, basically, who's about to actually sequence and actually generate the genomic information on a larger scale for the first time in human history. And he was working together with the George Church on the first public data set that's being produced by something called next generation sequencing, which actually means that you could sequence the whole genome compared to just doing the partial tests on the genome. And basically, during that process, he actually figured out that this was not particularly scalable process if you do it by hand, because it's computationally intense. You actually have to run it, rerun it. If you write it as a script or try to scale it on X machines and do it manually, that pretty much doesn't work. So we are going to tell more about that later on. But in general, we are actually focused on something that's called secondary analysis in genomics, which basically means, an example, if you have some tissue, let's say blood or saliva, and if you actually want to figure out and actually get some genomic information out of that, you actually have to prepare this tissue, and actually you have to put it in a machine called sequencer. And out of this sequencer, you actually get a small partial pieces of DNA or part of DNA, which actually don't answer on any particular question on basically if you want to ask, let's say somebody has a cancer and you want to figure out what mutation causes cancer, some rare genetic disease. So in order to actually get to that point, you actually have to run this data through various tools in order to actually get either some differential comparing this genomic information to some other genomic information, visualize it, etc. So, So in order actually to do that, you could do it also on one machine, usually, and just to give you the order of magnitude of the samples, just one sample can range between 80 gigabytes and 800 gigabytes, depending on the quality of the sample. So for something that's considered to be a cancer quality sample, you have to actually produce 800 gigabytes and process it to various tools in order to get information or just partial information on what actually caused the cancer. So what are you actually doing? So we are right now running on top of the Amazon Web Services, and I'm just going to mention two most prominent projects we are working on right now. One is for National Cancer Institute. We are building the Cancer Genomic Cloud pilot together with the Broad Institute and Institute of Systems Biology in the United States. And we are also working together with, and we are part of Genomics England project, where we actually are building the first functional graph genome of human, first graph genome of human DNA, which basically means that, for instance, right now, the most operations that are being done on genomic information and actually are being done on top of just one, imagine it like a flat line of information. So it's not particularly articulate or precise information when you actually compare it with just one sample, because basically you can miss the variations that's actually, you can spot in the largest species of humans, and actually, in order to actually do that, you have to completely change the paradigm and actually build a graph which represent various pets. And pets in that graph actually represent various individuals. So in order to detect better variations, structural variations, mutations, you actually have to compare against the population, not just one person randomly selected to be something we call a reference genome. And soon we'll also be running on a Google Cloud platform. Okay, what we're going to cover in this talk, so actually we're going to cover why is generally processing genomic information hard. And we're actually talk about parallel data access at scale. And we're also going to talk about the computation at scale using the researcher contributed tools. I'll give better explanation of each of those during the talk. So let's just focus on why, for the start, let's focus on why is processing genomic information hard. So there are many reasons, but we are going to focus on something we consider to be three main reasons of complexity of processing genomic information. So, as previously mentioned, the genomic data is quite big, and it's getting bigger. So, one assumption we have is basically based on the data of how many sequences are out there and how many data is being produced right now, is that only between 2014 and 2018, we're going to produce more than 2 exabytes of data. And basically, you can see that also on this graph is basically that with the sales of the NGS sequencers and basically the price drop of the sequencing operation, basically the amount of data is actually exponentially growing during the years. So, and also genomes are inherently sensitive. So, which basically means that security is also quite important part of the whole process of storing and processing genomic information, because this data is very personal. You can basically reverse engineer actually from personal information from genome information as well. And basically this data is also pretty much related to your relatives as well. So you cannot also fully anonymize information right now with the current paradigm. So you also, once actually all this data leaks out, you can't really take it back. So it's pretty much the same like with fingerprints. And actually one huge leak of fingerprints actually happened with the United States government a couple of months ago. So basically you have to store and manage this data pretty carefully. Also, one important thing is basically that the genomics research is highly collaborative and diverse activity. Basically, which means that usually it occurs and it's being done in large teams. So in this picture is just one of the teams that actually works on the cancer research. And this is the pan-cancer analysis of whole genome consortia. And this consortia has more than 700 researchers working on the same data set. And also you have to work with numerous analytical tools that are being built by the researchers all across the world. They're completely various. There is no standard, and basically can be written in anything or basically without any consideration for scaling or without any consideration for security, etc. So this is some those are basic points about why it's actually so hard to actually process the genomic information. So I would like to switch now to parallel access at scale. So why it's actually hard? So the thing is that when you actually, as we previously mentioned, you have to run your data through numerous tools. And basically, if you would, for instance, run this tool on a single machine, and you would want to process one single genome sample through something that calls standard whole genome pipeline that actually gives a differential between you and the standard genomic information called reference genome. So this actually can take anywhere between 2 days and a couple of weeks, depending on the size of the sample, if you don't parallelize this particular task. And also, this task, if you even parallelize them, they actually they have to access intermediary files during the various steps of this execution. So the common way how to actually solve this, and how pretty much everybody solves it right now, and we actually did the same thing, is to have the coordinator instance, which would actually have the NFS server and could be accessed by a multitude of the clients, and actually to store and actually share the data. Even though you can arrange this so there are no synchronization problems, and pretty much everything works relatively stable. NFS actually saturates at server saturates at pretty much 8 workers, because the network interface is actually the bottleneck of this setup. So this is good for certain tasks, but for some more complex tasks like more efficient whole genome processing or some population-wide pipelines that you run on top of our platform, this is not efficient enough. So how we actually try to address this problem? So we had a hypothesis that we actually, as we are living on top of the AWS right now, and we actually, while we were actually processing the data, we figured out that by pure empirical experiment, that when you're actually accessing the same data objects, and when you're actually trying to write, there are no the performance degradations we've seen with the NFS servers. Of course, so that was just an assumption. And we actually didn't have any proof for that before. So it would also be very convenient to actually present these objects that are residing in S3 as a file system, because all these tools, as previously mentioned, were not written with scalability or cloud resources in mind. So you would actually have to actually have to either download these files or move them around, but they actually had to be perceived as a local files. So we actually also assume that based on what we've seen so far, that cumulative read-write speeds should scale mostly linearly with number of workers, because if you're accessing S3 objects, you have this have your data objects actually replicated not only on the servers but also on the caching layer in front that stands between the S3 storage servers and actually you accessing that through S3 API. And also, the final and most important assumption is that total read and write speed on shared S3 objects would significantly exceed NFS server setup for more than 10 workers. So this is, if you just look at the diagram, basically, it's a simple illustration that there is no bottleneck, but this is not particularly important right now. So what was the challenge? Actually, the challenge is that none of the existing fuse-based solutions with S3 backend were either fast or reliable. There are a multitude of the actually file systems or implementation you could find, and actually that are open source that actually abstract S3 as a local file system and local objects, but performance would degrade very, very fast. And also, we have strict authentication policies. Basically, we can't really just mount the whole S3 bucket. And we can't just present you all objects, because you can't really access all the data that resides in one object. We actually have to implement very, very strict access rules, because, as we said, genomic information is very sensitive. And also, appending the data or random access or caching required rethinking with S3 backend. Thing is that, for instance, when you close the S3 object, you can't really add any information on further on S3 object. So basically, you would have to somehow abstract the whole idea of file into multitude of the objects. So, and also, you have higher latencies between the local file system and basically and compared to file local disk, and when you have the S3 between the metadata layer and actual data blocks. So this is the big picture of how it actually looks like in the practice. So SBGFS is basically sitting on top of the fuse, and it talks with S3. But for each access, it also talks with our service, which basically provides additional file metadata or authentication layer for all instances using the SBGFS. Of course, we are also aggregating the logs for audit, which is also important for us given the all compliance and security issues we actually have to cover. So this is how the performance actually looked in practice. So these are the pretty much same results we actually have seen with the NFS. There were no particular difference between the speed we achieved with the local NFS server and regular network access and actually accessing the data objects on S3. But more interesting part is basically that as we actually tested this on hundreds of machines, we actually had a cumulative speed up when actually compared to the NFS server access, and basically NFS server would completely break down around 20 instances. Just to go back, basically, this actually red line is also interesting. When we actually implemented also the local caching, where we would actually cache the files once they would reside on the worker machines, we would even achieve greater speed because some tools would go again actually access the same file or same file blocks. So this is the one of the interesting problems we had to solve, and we are still solving. Basically, how to scale the data access on such a level of data sets. So just also to give you an illustration of this complexity and size of data sets we are working with, we also, in collaboration with NIH, brought the complete TCGA data set, which is actually the data set that's most sophisticated, most complete cancer data set you can find in the world. And basically, it consists of 1.5 petabyte of various research data, sequenced genomes, pictures, whatever, and it's actually expected to grow 2.5 more petabytes in just next year. And also to process this data, so how you actually do it. We are not writing our tools, and this is very important. So actually, for someone to actually trust the results, you have to run it through tools that have been confirmed by community of researchers and academia. So you can't really write your own aligner or any of the tools and actually expect somebody to trust it or believe that the results are correct. You actually have to integrate the tools and algorithmics that's been developed, that's gone through a review process and basically is considered to be correct or correct to a certain degree. So just one computation doesn't take one tool. This is just one anatomy. This is a part of something that we call genomic pipeline, which is nothing more than a directed graph of the tools and their inputs in order to actually just get from the raw data to certain biological answer. This is one of the more popular pipelines. This is the whole genome pipeline, which basically takes your genomic information and spits you out the difference compared to certain reference genome or other person that you can use for further inspection of what's actually going on with the genomic data or what are the treats you're interested in. So we could actually go many ways. So why we then use, for instance, SunGrid Engine or Hadoop or OpenMP or Spark or Mesos. One important thing to have in mind, we actually started to implement our platform 5 years ago. And actually, while you can actually implement various algorithms and very nice things on top of the Spark, we actually use Spark, and we can spawn the Spark clusters. But to actually embed tools that had been written by the academia, you need to impose more strict control on how you actually treat these tools and how you parallelize them and how you actually operate on top of them. Moreover, you can't really trivially split the data. You cannot just take the genomic data and split it on 100 of instances and expect to get a correct answer by just treating the partial data. So basically, Hadoop was not particularly useful for us, and basically we had to implement our own paradigm and our own platform. And this is very, very high-level diagram of our platform. So we basically orchestrate the particular tasks and have dedicated services that actually monitor the tasks. And if something fails, and in the cloud, and especially in the Amazon cloud, things are failing all the time. So you actually have to retry doing the things. You have to watch whether some parts of the task fail, restart them, rerun them, actually be sure that something failed. So it's not particularly easy to do. So also, to give you some context, basically, if you want to join this game, you actually have to work with more than 10,000 bioinformatics tools. And just as you can see in the picture, this is usual number of tools for more complex pipelines is anywhere between 30, 40 to 100. And for instance, this is one of the more popular cancer pipelines, and it has around 50 tools in it. So how do you actually do that? So in the beginning, we actually start to make a beefy image on Amazon, and which would actually consist a bunch of the dependencies and tools packed for EC2, where you could actually run the instance and have the tools on that image. We then switched again to actually just having only dependencies by dynamically actually putting the tools on top of the instances, but that didn't scale past 200 or 300 tools. Because the problem is basically you're not only dealing with the tools that have different dependencies, different versions of libraries that they actually should and have to see, but you also have to provide the different dependencies for the different versions of the same tool. So we actually started to package everything in Docker containers. So how does it work? Basically, we have very simple image nowadays. It actually just grabs the containers with the tools that it needs for actual execution. And everything is wrapped through something that's called Common Workflow Language. So now this is something that is becoming a standard in our industry, and we are actually also participating in the design of this particular, as you could call it, a language. It's pretty much a similar like W3C for the web. Basically, there is a consortia that actually tries to integrate the standards, and so you can actually port these tools across the platforms, which basically means if you have CW wrapper with a Docker image, you can run it on our platform, or you can just take it somewhere else to any platform that supports the CWL, or run it locally, which is quite nice if you're actually trying to develop the tools and actually then just get them to work on scale on our platform. So what were the benefits of using the Docker to deploy the user-contributed tools? So Docker actually enables a solid resource isolation at the container level, which basically means that you not only can isolate the files that the Docker container can see, you can isolate the network, you can isolate the processes to the namespaces. This is particularly nice because you're not in control of these tools. Also, simplifies the deploying and managing tools at scale. Which means that, as you can see, when we were packaging so many dependencies on a single image, this started to break at a certain point. Also, you can move your Docker container actually to any platform that supports it. It's becoming widely adopted standard for the containers, and basically you are not relying on SBG or any other platform. And also you can work locally. As said, you can basically package everything in Docker container, work locally, and just basically deploy the container on a platform using your SDK. So it's not all that simple. So, Docker comes with a price. And basically also, there are security risks that you actually have by just using the Docker. What are the most common security risks that we've been seeing so far? Basically, Docker is running under root privileges, which means if somebody manages to exit the Docker container, that person can actually gain the root privileges on that particular machine. And users can also intentionally or unintentionally post the malicious app store platform, because you could pretty much wrap anything and post it as a tool on an SBG platform. And also, if the resource management is not properly configured, applications could actually do the damage outside of the container itself. For instance, if you actually let the container to see the files that it shouldn't see, or files that are residing from some other execution, it's not particularly a great situation that you're dealing with. So how we have been able to secure Docker containers further? So we actually isolate the data access, so the Docker containers can see only the data that they should see. There is you don't have to let Docker containers see whole file system or share the same data with the other Docker container unless it's completely necessary. You can also further isolate the network resources for each container by firewalling the particular network interface associated by that container. And also, one nice feature of actually Docker is that you can always combine the Docker containers in a more, complicated structure, but you should be careful when you're actually linking the containers. And of course, you should always aggregate log, and so you have them for any forensic activity you need afterwards if something bad happens. So basically, how do you actually optimize for computations? Also, one important thing to say is that with each tool, you can describe what are the resources that this particular tool requires. You can say, for instance, this data that enters this tool can parallelize by using this algorithm, and basically then it can run, it requires one core, etc., etc. So you basically can describe on a tool and what we call pipeline level, how you actually can parallelize this pipeline. So as long as you actually say to our platform how you actually want to run it, we are able to parallelize it on hundreds or thousands of machines. So that brings us to general takeaways. So what we learned so far is that basically you should understand the data and problem you are solving. For instance, as previously mentioned, it took us a little bit of time to actually understand that neither of previously existing paradigms didn't really solve our problem, and we actually had to build our own platform to solve something like that. And we are 5 years into development, and we are still working on that, and we don't see really the end of that. Also, you should understand the scale and the nature of data. It's not really the same thing dealing with gigabytes of data, terabytes of data, petabytes of data, or even more. And basically to understand how people actually share the data, and because you always want to bring computation to data, not the other way around, especially on the larger scale of data. Also, one thing is to leverage infrastructure you're working on top of. For instance, we could ignore the fact that we are on AWS and just treat it as a little bit more dynamical local environment. But by using the S3, we've been able to actually leverage much higher speeds and much higher scalability than we could actually do with just a regular NFS server. And the most important thing is to measure everything. Unless you actually measure whatever you're working on, you have no info, you have just only your intuition. Intuition is pretty crappy tool to actually have. So that's pretty much it. I'm sorry if it's a short talk. So please join us if this is an interesting topic for us. We are hiring, and you can find the list of our jobs at this site. So thank you. And if you have any questions, please go ahead.